are interested in learning more about today. We are always excited to welcome new researchers into the folds of the alternative protein scientific community. So very delighted that you're all here with us today. Today, I'm joined by two very brilliant scientists, Dr. Elliot Swartz of the Good Food Institute and Dr. Roni Rak of the Agriculture Research Organization in Israel, to discuss the very rich and impactful opportunities for cell biologists to get involved in the alternative protein field. We are at a pretty critical juncture in the arc of our food system, and we view cell biologists as absolutely essential in this quest to preserve and to restore human, animal, and planetary health. So in just a couple of short moments, you'll learn a lot more from Elliot and Roni about the scientific case for alternative proteins, research opportunities in this emerging field, and resources we have to support your work. If you have any questions throughout, please feel free to drop them in the Q&A section, and we'll make sure to address them. And then one last item before we get started is that I would love to invite you all to join the Alternative Protein Researcher Directory, which I will drop the link to in the chat shortly. GFI's Alt Protein Researcher Directory will allow you all to share your work on alternative proteins, publicize the ways you hope to collaborate with other experts, labs, and companies, and identify potential partners to help you scale up the impact of your work. If you're compelled by what we share today about pathways for cell biologists in this field, join so that you can receive curated updates on research developments, funding opportunities, scientific events and forums, and collaborators that match your skills and interests. Alrighty, so without further ado, let's dive in. I'll pass the baton over to Dr. Elliot Swartz to get us started. Thanks for being here, Elliot.
All right, thank you, Amy. And please shout at me if you can't hear me, but I'm assuming otherwise that I'm on. So welcome, everyone. Thanks for attending. My name's Elliot. I am a principal scientist focused on cultivated meat technology at the Good Food Institute. And today I figured I'd be giving a bit of an overview about some of the interesting cell biology, cell line related aspects of cultivated meat. And hopefully that will spur some thinking in your head about how you might be able to contribute to addressing some of the challenges in this interesting industry. So before I begin, I just wanted to share a little bit more about the Good Food Institute, or GFI. We are an international network of nonprofits that are developing a roadmap for a more sustainable, secure, and just protein supply. And so we have teams spread out across the world in the United States, Brazil, India, Israel, Europe, and the Asia Pacific region. And all of our programmatic work happens under the same umbrella of trying to advance these alternative protein technologies in the marketplace and ensure their success. And so we're very critical in helping to ensure that there's really an innovation ecosystem in place that will drive these technologies forward so that they can have impact in the future. So the question that we're really interested in addressing is, how are we going to feed a growing population, one that is expected to near 10 billion people by the midpoint of this century? And the core challenge here is that the way that we produce meat today has numerous negative externalities associated with it. And so this is only expected to become more and more of an issue as meat consumption increases over time. And the UN predicts that meat consumption may increase anywhere between 50 to 100% by the midpoint of this century. So meanwhile, animal agriculture is already a top contributor to water use, air pollution, deforestation, loss of biodiversity. It accounts for around 15 to up to 20% of global greenhouse gas emissions, depending on how you account. And it's responsible for about 3/4 of the world's agricultural land use, and about 3/4 of the human medically relevant antibiotics are given to animals rather than humans, such that it's a cause or driver of antimicrobial resistance. And then finally, another public health threat is zoonotic disease emergence. And so it's quite likely that the next pandemic could come from a farm that raises chickens or pigs, et cetera, as has happened in the past. And so all of these negative externalities, we think, can really be addressed through alternative proteins. And so our theory of change is that if you're able to develop these food products from different means that compete on price and taste with conventional animal meat, then you can win in the marketplace without having consumers to have to make a large behavioral shift. And so similar to what we're seeing with electric vehicles, where finally the price points are coming down to be more competitive and the performance is increasing such that consumers' needs are being met, you start to see inflection points in the adoption of these new technologies like electric vehicles. And so we think a similar trajectory can happen with alternative proteins. And we define alternative proteins by products that can be created from plants, derived from fermentation processes, or cultivated from actual animal cells, which is what we'll be talking about today. So cultivated meat is real, genuine animal meat or seafood that can replicate the sensory and nutritional profiles of conventionally produced meat because it's really comprised of the same cell types that are arranged in the same or maybe similar three-dimensional structure as animal muscle tissue. And so you can see some early stage prototypes from a variety of different companies in the space, spanning seafood and other meat products. And you might be aware that as of last week, there are 2 companies in the United States that received full clearance for selling products to consumers. One of those was Upside Foods. And each of these products went under FDA evaluation for over a year, where FDA concluded that these are really no different from other animal cells with respect to safety for use in food. And so this was a pretty big hurdle for the industry to cross since its inception around 7 years ago, going from 0 to now entering the marketplace for the first time in the United States. So at a high level, the production process starts by taking a sample of cells that can be obtained from the animal, and those cells are taken back to the lab where they're subcultured and derived into cell lines that are workable for upscaling into a bioprocess or for end product development. And the goal here really in the first phase is to create a large amount of cells. So you're proliferating cells in consecutively larger bioreactors to create the biomass that can either feed directly into these products as individual ingredients or potentially move downstream into a process where they're differentiated, sometimes in the presence of a scaffolding structure for more structured products. And eventually these cells or these tissues are harvested and then formed into a variety of different final products using methods and tools that are already widely used in the food and or meat production industries. And so since this is an audience of cell biologists, I figured I'd focus mostly on the upstream portion of this because cells are really essential to this entire process. They are, the major primary input from the beginning. And there's a lot of interesting questions and challenges that can be addressed through cell biology. So just to dive a little bit deeper into the cell line procurement stage here, typically what happens is a small biopsy is taken from a living animal, or in some cases, an animal that was recently slaughtered, and those cells or tissues are still viable. So, as you can imagine, you're creating meat, so you might want to take muscle tissue or a fat tissue sample. In some cases, a fertilized egg is used, a variety of different possibilities that we'll talk about in a few slides. Those tissues can be taken back to the lab, broken down, and then sorted for the cell types of interest, and eventually developed into a cell line that can propagate in culture. Or in some cases, some companies are actually using sort of primary cells where they're rebiopsying from an animal repeatedly and growing those cells up in independent batches. But nevertheless, you want to do some form of characterization for these cells to ensure that they're going to perform well in your process. So looking at things like growth rates, metabolism, different types of omics analyses are quite typical in the R&D process upstream in these companies and in labs. And then finally, these cells are banked in master and working cell banks that look quite similar to what you see in other animal cell biomanufacturing industries like biologics or vaccines. And so one of the interesting questions with cell line procurement is that really the health of the animal is an important downstream indicator of safety. And so one of the most important things you can do upstream from a quality control and quality assurance process is to ensure that you're obtaining cells from a healthy animal. And one of the other interesting questions that the industry, I think, is now starting to breach relates to, whether this animal was living and slaughtered and what downstream implications does that have on certain religious certifications for the end product. So we actually surveyed around 40 different companies last year that responded around all these questions related to cell lines, and we published this report just a few weeks ago. And so I'll share some of that data throughout the next couple of slides. But you can see that companies in the industry right now, about half of them acquire cells from recently slaughtered animals, although I think the desire is to shift to acquiring them from live animals so that slaughter isn't involved in the process. But sometimes it's hard to get your hands on, biopsying tissues from living animals. It's easier to partner with a slaughterhouse or abattoir. However, this is also important for things like halal certification, where slaughter is basically involved in the process from the get-go. And so many companies have a strong preference for these cell lines complying with halal certification, especially dependent on where they sit regionally or geographically. So that's an interesting question that is being, I think, asked in the industry. Same thing with kosher certification. How does this work with respect to cell lines and other media inputs? So you might be asking, what cell lines or cell types do companies and researchers use in this field? And it's essentially any type of cell that can differentiate into the components of meat. And so, you typically want to create muscle tissue, fat tissue, connective tissues. And there are a variety of different adult stem cells or even pluripotent stem cell types that companies are exploring for this use. And so you can see the breakdown here. There's really no majority for the selection of cell types that are being used in this industry. We really haven't seen any consolidation yet because there's just so many different approaches being taken, and pros and cons of each, from, starting from an adult stem cell versus an embryonic or pluripotent stem cell. So you get interesting pros and cons that break out, and some people think that certain cells are advantageous versus others. When you ask companies what tissue types they're creating, it's perhaps unsurprising that most of them respond that they are interested in creating skeletal muscle, fat, and also fibroblasts or connective tissues. But you also do see a handful of companies exploring the use of endothelial cells or other vasculature, presumably probably to make more structured products, as well as other tissue types, whether it's cartilage, bone, other organs like cardiomyocytes or hepatocytes to create organ meats like foie gras. So one of the interesting questions, from an R&D perspective is, what are the desired attributes that we want for a cultivated meat process, a cost-effective one? And we ask companies, what sort of characteristics would you look for in a cell line? And you can see that companies are really interested in high proliferation rates with low doubling times. This is sort of self-explanatory. It can speed up your R&D process as well as have cost implications, especially at scale. You want genomic stability across multiple generations, important from a regulatory perspective and a reproducibility point of view. Immortalization, which we'll talk about. So cells that, sort of divide indefinitely, usually easier to work with or bank over time. One of the other interesting areas is that, a lot of these cells, like muscle cells or fibroblast cells, are typically adherent, and so they need a surface to grow on. But this usually isn't, compatible with how we think about scaling up animal cell cultures, which are typically done in suspension. And so transitioning these cell lines to adapt them to suspension culture is often a challenge that many companies face early on. Similarly, differentiation, obviously important. Metabolic efficiency, I think, is actually underrated here. I would put it near the top of my priority list to think about from a cost and environmental impact perspective. And then other things that are related to the sort of process, especially when you get to scale, shear stress, as well as the buildup or limitation of metabolic byproducts like ammonia become increasingly important as you try to grow large quantities of cells. One of the other areas of interest for R&D in this field is to eliminate the use of animal components in the media and throughout really the entire process, aside from the cells themselves. And so serum-free media creation is really important, and also somewhat of a challenge because we have to create serum-free media that span all of these different species, from crustaceans to fish to avian species like chicken. And a lot of that hasn't really been done before. And so a lot of this is all new in terms of media optimization and culture conditions. But nevertheless, you can see that a lot of companies have had success developing serum-free media. About half of these companies say that the performance is relatively equal to serum-containing media, while some say they have eclipsed the performance of serum-containing media. So that's a good sign. While other earlier stage companies are most likely still in the development phase of switching and or increasing the performance of their serum-free media. So another area of interest is immortalization and engineering of cells. Different types of immortalization methodology can be selected. And so here you can see a breakdown of immortalization methods, from spontaneous immortalization, which is the sort of leading method of choice for companies for R&D and commercial purposes, presumably because this sits outside of the boundaries of most genetically modified regulatory pathways. You also have non-integrating expression, which sits about in the middle, and then permanent genome alteration, which is the lowest preferred method, presumably, again, due to this requiring a longer or more rigorous regulatory pathway. And if you ask companies about genetic engineering methods for R&D or commercial use, you can see that about half of companies aren't even really thinking about using genetic engineering. So they're just doing sort of a GM-free process. Others are looking into it for R&D, and then others, a smaller percent, around 20% of companies, are exploring this for commercial use as well. And in fact, one of the companies that was approved for sale in the United States last week, Upside Foods, does indeed use genetic engineering to immortalize their fibroblast cell lines by overexpressing the chicken telomerase gene. So I think this opens the door for a lot of people exploring this as an option, especially in the United States. So this is just a small snapshot of companies. There's obviously, there's now 100 cultivated meat manufacturers around the world, with another 50 to 100 companies that sort of play a horizontal role in the value chain, sort of specialist companies, some of which I've listed here just so you can get a sense of some of the type of work that goes on related to cell biology upstream. So there are a few companies like Roslin Technologies and Oppo in New Zealand that are creating cell lines specifically for use in this industry. So to sell validated, sort of characterized cell lines to researchers in academia and also for commercial use. You also have companies like Triple Bar Bio that uses microfluidics technologies to enhance cell line development. So thinking about, can we speed up the time it takes to create a suspension adapted cell line? This is something that a company like this can help with. Prolific Machines is a startup that's trying to engineer and grow cells entirely without growth factors. This is really important from a cost perspective. And then finally, you have, obviously the culture conditions are very dependent on the cell culture media that's being used. And so you see companies that are really interested in metabolic engineering, modeling, et cetera, to inform media development and to optimize the feed conversion ratios. So to ensure that, the ingredients that are in the cell culture media are exactly what those cells need. And you want to limit waste because cost for this industry is really, really sensitive. So hopefully that gives you a sense of, some of the activities and challenges or things that are going on in the cultivated meat industry. Maybe you can relate some of that to the own work that you've done or that you're doing right now. And just wanted to share a little bit about my path and how I got here. So I majored in neuroscience as an undergrad and also have a PhD in neuroscience at UCLA. But I would actually consider myself more of a cell and molecular biologist. And so for my graduate studies, I worked with human iPS cells to model neuromuscular disease, making skeletal muscle tissue that you can see here, sheet of skeletal muscle tissue contracting. And then the sort of end goal of my project was really to try to create a spinal cord in the dish. So you see an iPSC-derived group of motor neurons that are attached to these iPSC-derived skeletal myotubes, and these cells are derived from ALS patients, trying to get insights into how this neuromuscular disease might have happened. And that now I'm a scientist at GFI, where I've been for the past 5 years. But really what led me to transitioning my career path from something that's more applicable to, let's say, drug discovery or therapeutics for neurodegenerative disease, which was the focus of my research. I really came to grad school with the hope to go into regenerative medicine. But throughout my time there, I became more and more concerned and interested in addressing climate change, which I viewed as probably the biggest issue of my lifetime. I learned about cultivated meat back in 2015 or so when some of the first companies were being formed. But back then there wasn't a lot of information. And so I actually wrote a blog post online, and a lot of people found that. I started getting cold calls from investors. People found it useful. And so that really convinced me to, I think, really kickstart a career in this field while it was still young, so I could sort of grow with it. I found out about GFI around that same time. It was founded in about 2016, 2017, our organization. And then finally, when I joined GFI, I became vegetarian, so I could sort of completely walk the walk if I was going to be out here telling everyone to reduce meat consumption and coming up with ways to solve this challenge. And so there's this term ikigai in Japanese that sort of unites all these different things around your passion, your profession, your vocation and mission, and being able to marry these under one umbrella. And for me, I think that's sort of the cultivated meat field, because it addresses really important things about what the world needs and what I care about from a mission perspective with respect to animal welfare, climate change, human health issues, food insecurity. There's also really challenging scientific questions. So this is certainly by no means easy to do this, and I think it's going to take a lot of time. But obviously I can leverage sort of my training as a cell biologist to get into the field from the beginning. And also you can get paid for it, obviously. So I have a job, and hopefully you guys might consider a job as well in this industry. There's more and more opportunities every day. So thank you for your attention. And I think I will pass it to Roni now.
Hi, everyone. So I'm really excited to be here. I'm talking from Israel, so it's late evening now. And I want to say first, very thank you for hosting me. And I want to say a lot of thanks for the GFI, also for organizing and get me into this event, and also for the huge support in my research. And I'm a new researcher at the Volcanic Institute, the agriculture research organization in Israel, and my lab is working on cultured meat. So this is the first lab in Israel that studied cultured meats, and this is the whole purpose of my lab. I want to tell you a bit about my academic journey, because it wasn't trivial, and it was very surprising for me to get to the cultured meat field. So I did my PhD in Tel Aviv University, where I studied cancer and designed novel drug inhibitor for cancer. The first postdoc I did on a very, very different subject was linking tRNA expression to longevity, studying it on C. elegans in Oded Rehavi's lab. And my second postdoc was dealt with also translation and tRNA expression, but here I a bit combined the previous two research and studied how tRNA expression and protein translation affect proliferation and differentiation, and how it changes between cells that proliferate and cells which differentiate, which are the two different aspects of cell life. After the second postdoc position, I applied for a position in the Volcanic Institute, which was designated for studying cultured meat. I thought that I might be able to combine all the knowledge and all the expertise in the research I gained and study cultured meat. I have to say here again, thank you to GFI that I consulted with them a lot about how my future lab can look like and what are the hot topics in the cultured meat area right now. So I talked with the GFI and I thought, how can we feed the world? So it's not very easy yet. So we're not here yet in just thinking about food and making it just magically appearing on our plate. There is still a lot of work to do. And I thought there might be a lot of research opportunity in this area. I will skip on what is cultured meat because Elliot presented it very nicely, and go to my passion and how I see research combining in this aspect. So during billion of years, cells evolved to become from unicellular organism to multicellular organism. It took 2 billion years to get from a unicellular organism to a multicellular organism. I thought that the cultured meat area might present the opposite. So taking multicellular organism and adapt it to grow independently and become something like a unicellular organism and have the challenges of growing alone in a rich environment. So when we gain the multicellularity, there's a lot of profit in that. So there is cell specialization. We can acquire a lot of new traits such as better immunity, muscle, so we can be stronger, we can digest much more, we can move between environment and sense the environment. We can also get to a higher size and higher diversity in the organism type. However, unicellularity has its own benefits. So one cell can be self-contained. It can have all the traits he needs, all the it can fulfill all his needs and sense the environment. It has much fast reproduction rate, so it actually can adapt better and colonize better environment. Also, it demands less resources. So to create better cultured meat, we have to get back the unicellular properties we actually lost in the evolution. In addition, multicellular organism carry a lot of a huge genome, a lot of other genes that are not relevant anymore for the cultured meat production. So in multicellular organism, there is dichotomy between cellular proliferation and cellular differentiation. The cells can actually, when they proliferate, they have a lot of cell autonomous function. Most of the proteins that are expressed are protein related to cell cycle, to translation, to transcription, and cell autonomous function. However, when they differentiate, they express a different set of genes. The genes are more related to tissue-specific and cell communication, and different panel for each type of cell. When we want to move between those axes, we have actually, if we go to the proliferating cell, we have all kinds of stem cells, induced pluripotent cells, or cancer, if it goes wrong. And when we talk about which is the good cell source for cultured meat, as Elliot already presented, there might be a very different source for cultured meat. However, there is a trade-off between proliferation and differentiation. So cells that proliferate well might be very hard to differentiate, while cells that are more differentiated, they also proliferate a bit less. So my lab is actually studying the genetic regulation of proliferation and differentiation. We chose to work on mesenchymal stem cells, which are a bit in the middle of this trade-off. So they do proliferate and they can also differentiate, but there is still a lot of way to improve both the proliferation and the differentiation. So we are working on adipose-derived bovine mesenchymal stem cell animal, either alive and by biopsy without hurting the animal too much, or from a slaughtered animal. It's interesting. I was discussing people from religion, rabbis in Israel, when they actually much prefer taking it from a slaughtered animal, a kosher slaughtered animal. I actually think it's not good to slaughter animal for that, but it's still debatable. So the cells we're actually producing are mesenchymal stem cells. They have a fibroblast-like phenotype, and we can identify them by a lot of characterized surface marker identified by FACS. We can also isolate and cryobank those cells, and they maintain their proliferation and differentiation in culture. So the first, we isolated the cells from bovine adipose tissue. Oh, sorry, it jumped. We measured their growth rate, and we wanted to make sure they're stable toward many passages in culture, and we can see that they maintain their doubling time of about 36 to 40 hours throughout the passages. And we also identified the surface markers by FACS. Most important is that the cells can differentiate into the desired tissue, and to check their multipotent potential, we can actually differentiate them both to adipose tissue and also to chondrogenic tissues. So how can we look at the trade-off, and how can we optimize the proliferation and differentiation of those cells? So my lab is actually focused on this area of how can we can adapt them genetically. Our genetic adaptation is actually a tool to study both what can we do genetically, but also to identify other small molecules or inhibitors that can affect the process and optimize the proliferation and differentiation rate. How do we do that? We want to see how the evolution actually changed and shaped the destiny of the cells, as we talked about multicellularity and unicellularity, and we want to apply the evolution forces of to study those aspects. We started first in looking at natural variation and see whether different type of animal can contribute to the phenotype of fast growing, fast proliferation, and efficient differentiation. And this project is actually funded by GFI in Israel. The other way for us to study these aspects are using genetic editing variation. So for this purpose, we created a whole genome library of CRISPR knockouts and introduced them to the mesenchymal stem cells. I will expand more about this project, which is funded by GFI worldwide in the initiate.
All right, it looks like Roni's internet may have cut out. Hey, Roni.
Hi.
We are experiencing just intermittent Wi-Fi blips, I think, on your end. So you just froze for about 30 seconds, but it's possible if you turn off your camera it might work a little bit better and we'll get a steady stream of audio.
Yeah, okay. So I'm stopping the video. Do you hear me better now?
Perfect.
Okay.
Thanks, Roni.
So I'm starting from this slide again. Okay. So to study the genetic basics of proliferation and differentiation, we wanted to apply the evolution forces on those cells and to study how the evolution actually might affect those processes. So first we used natural variation of animals. So we can use different type of bovine that was selected to be efficient in meat production or in milk yield. So they have different natural variation, and we can see how those natural variation affect proliferation and differentiation of the mesenchymal stem cells. This project is funded by the GFI Israel. The second project is using genetically editing variation. So we use CRISPR, we design and apply a whole genome CRISPR screen that targets each of the genes in bovine. And we can use artificial selection to mimic evolution forces to select for phenotypes that are more adjusted to the cultured meat industry. This project is was funded by the GFI initiation grant program. So using those two approaches of natural variation and genetic editing variation, we can measure how the genetic changes affect growth and environmental growth condition, and how it can actually adapt to less use of growth factor, different type of scaffolding, and all other processes related to cultured meat production. So I want to expand a bit more about the CRISPR knockout genetic variation. So we identify how can we select for genes that might affect the proliferation rate or differentiation rate. We analyze the DepMap database, which have CRISPR screen data of over 1,000 cell types. And we can identify which genes actually has the best effect on cell growth. So each gene gets a score based on how it affected the growth of all of each individual cell type. And we can see that the different genes get the highest score. It means that when they are knocked out, the cells that had this knockout actually proliferate much better. We selected a specific gene for a proof of concept. We focused on the P10 gene. Yeah. Okay. And when we knocked out P10 in a bovine mesenchymal cell, we actually saw an increase of 20% in growth rate. 20% is maybe sound a bit not a lot, but when we calculate cell growing to 30 days, we can see that the number of double change from 23 to 29 cell doubling. And when we calculate the weight arising from this cell growth, we can see that we increased the number from 10 kilogram to 600 kilogram just by increasing the cell growth by 20%. And next we applied the whole genome CRISPR screen to identify more candidates and to actually characterize the genetic background of the cells and to see which genetic pathway can influence those cells. Again. Okay, so the first step was to generate CRISPR Cas9 expressing cells. And we were able to do that in bovine mesenchymal stem cells. It wasn't very easy to calibrate the viral transfection of the cells, but we were able to do that eventually. And then we did appropriate selection pressure. So we can use the whole genome CRISPR library to study many different effects and to characterize which genetic pathway and genetic regulation affects the growth of the proliferation rate, the differentiation potential, and also studying the condition, for example, growth in suspension. First, we started with proliferation rate, and we can see that once we introduce genetic modification to each cells, we can actually select the cells and identify which cell contribute to their rapid proliferation. So we did that experiment and followed the cells and sampled them in individual days. So for example, day 1, day 10, day 20, and day 30. And we can identify using next generation sequencing which genetic modification contributed the most for the proliferation rate. When analyzing the data, we can see that several genes increase in their abundance in the population during time. For example, this gene, which is not a new gene, it's P53. Also P10 increased in the population, showing that knockout of this gene actually contribute to cell proliferation rate. In addition, when looking at the library of the genetic modification, we can see that over time, the proliferation rate increases and doubling time decreases. And also, just looking at the population, we can see that there is an increase in cell adaptation to the medium. I will stop here. So this is my lab. I want to say thank you to all my students and thank you all so much for hosting me. I hope the internet didn't fall too much.
No, that was fantastic. Thank you so much, Dr. Roni Rak, for a fabulous presentation, and also to Dr. Elliot Swartz for your presentation as well. All of you in the audience, if you haven't had the chance to do this already, please do add any questions that you have for our speakers today in the Q&A section of your Zoom bar, and we will get to them shortly. In the meantime, I have a few calls to action for those of you who are interested in cultivated meat, interested in continuing to explore your career as a researcher in this field. The first is to join us at gfi.org/researchers and join the Alternative Protein Researcher Directory. Again, this global researcher directory will allow you to share your research, publicize ways you hope to collaborate with other experts, labs, or companies, and identify other collaborators and potential partners to help you scale your work. You'll also receive a bi-monthly newsletter. Thank you. Next slide. We also invite all of you to explore key research opportunities at gfi.org/solutions. So as is abundantly clear from hearing about some of Roni's research and about some of the opportunities that Elliot outlined, there are so many opportunities and unanswered questions when it comes to cultivated meat science that we need your brains for. So if you are interested in kind of exploring a high impact career, curious about what research bottlenecks exist, this is the right place to go for you. And then lastly, I also invite all of you to generally check out on the next slide, the many open access resources we have for researchers at gfi.org/science, including the Alternative Protein Literature Library, our State of the Industry reports. We have a research grants program with our RFP launching very soon, later next month, and some deep dives into the science of plant-based cultivated meat and fermentation. So again, those of you who have questions, feel free to drop them into the Q&A section of our chat here, and we will get to them shortly. But thank you all for your participation. Alrighty, so I do see here a question from one of the members of the audience, wondering, I think this is a question for Elliot, if you can speak a little bit more about the use of iPSCs for cultivated meat and maybe focus particularly on the regulatory landscape and any hurdles you might anticipate.
Sure. Yeah, so it's an interesting question and one that has come up quite a bit for people that are familiar with pluripotent stem cells. So I'd say, as you saw in the survey data, there are companies that are using iPSCs. And, as Roni discussed, the advantage here is that, they proliferate quite quickly. Generally culture conditions are defined, although they have to be tweaked, depending on the species that you might be working with. We've never really generated iPSCs from a variety of different like crustaceans or seafood related cell lines, for instance. So that's an interesting challenge that some researchers are having. But the challenge is getting them to differentiate into the cell types that you want. And so a lot of companies, in addition to how it relates to regulatory, is twofold. So one is in the actual method of generating the pluripotent stem cells. So this is not like an easy, black and white answer. I think it really depends on where you are in the world. So for instance, Europe might classify like even the use of, let's say, an mRNA to transform your cells would qualify as a genetically modified thing. So any sort of like nucleic acid that enters the cell might trigger GMO regulation in Europe. But that's not how we understand it to occur in the United States and in some other places in the world. So you can use things like Sendai virus or other, non-integrating viruses or methodologies to generate your iPSC. So that's generally the approach that companies will take. And then from a differentiation standpoint, you see a lot of engineering there as well. So, overexpression of MyoD or some other master regulator for those cell types of interest, whether it's fat or muscle, those can really control the differentiation process into what you want to do. But of course, then you're using in most cases, a genetically engineered construct within those cells. So it's a challenge from a regulatory perspective.
Thanks, Elliot. My next question for Elliot and Roni both, and this is kind of admittedly a big question, is it's very clear that we're at this exciting time to be entering the alternative protein field because there seem to be dozens of opportunities cropping up on the daily, but it's also maybe a lightly nerve-wracking time to be exploring careers in this field because cultivated meat and alternative proteins center on emerging technologies that don't have well-defined career pathways like there are for more conventional and well-understood paths like biopharma. So I'd love to get your perspective, Elliot and Roni, on what you think of as the major education and training dimensions that helped prepare you from where you are today, so that those of us in the audience that are, there isn't a cultivated meat major that they might take, but what would you say are the foundational kind of skills and experiences someone should try and amass, maybe in a more informal capacity or in a formal educational environment to prepare themselves for this sector?
So again.
Yeah, Roni, do you want to start us off?
Oh, my internet is so unstable. So sorry. Do you hear me?
Yes, you
Okay,
sound great.
So I wanted to say first that from a scientific point of view, the cultured meat is very unexplored and it's fascinating. The thing that attracted me most to the cultured meat area was looking at it as kind of new organism that emerged to the world. And I think it's a new organism that is going to feed the entire world population, in my perspective, and it's not researched enough. So there's so many opportunities how to use the cultured meat as a research tool and to study what is going on with this new organism. So this is what actually attracted me as a scientist to the cultured meat area. Of course, that since it's not yet very advanced, so we also need to contribute it to become the world fitting opportunity. So that's my point of view for that.
Yeah, I mean, I'd say one thing I didn't mention is that I'd say, cultivated meat is like one of the most interdisciplinary fields that I can actually imagine. You have upstream, you have cell biology, you have people that are, working in chemistry and biochemistry related to media inputs. You have engineers bioprocess engineers, mechanical engineers, chemical engineers coming together to help scale the process up. You have food scientists, coming in at the end stage, and you have a blend of all of this sort of different disciplines coming together. And you really need success in all of these areas to meet the cost requirements that we're going to need to commercialize these products. And on top of that, you are intersecting and getting more, in tune with, environmental sciences, or how these issues intersect all these other global challenges. And so I think with that said, as Amy mentioned, there's, few programs, although we expect and hope to see more programs that are specifically tailored to education for cellular agriculture or cultivated meat. But really, no matter what you did, I mean, I think if you're an engineer, if you're a scientist, and even if you're outside of ththose disciplines, you can find a role in this field or in other alternative protein fields. Policy work is interesting too, and a lot of the scientists that do science by policy can find a home here as well. So, I mean, yeah, I'd say that opportunities are boundless, and it's exciting to get into something that's new, so that you can really grow and carve out your own expertise and role within the field before it becomes too much of a web, I'd say.
Thank you both. Yeah, I appreciate you speaking to the reasons someone should choose a career path like this one that's relatively uncharted compared to other more conventionally and well understood paths. I think there's a really compelling case there to be part of this massive paradigm shift towards a better food system. I know as a lot of folks consider their career paths, career security is a big dimension that often comes up. So kind of along this same vein, I would love to hear from Elliot and Roni, what are some of the signals you've been seeing that this might be part of a durable technological or paradigm shift with real disruptive power, and not just another technology that overpromises or like comes and goes, and thus not something that somebody might want to focus their long-term career trajectory on?
Well, I can start by just saying that, I've been at GFI for 5 years, and so I've witnessed some of the growth within this industry sort of firsthand. And I think one of the exciting things is that, which I alluded to, but you have all these different companies that are not cultivated meat manufacturers, but they might be other life science companies doing things in cellular therapeutics or regenerative medicine, other aspects of biomedical research, or even other fields. And, part of that business model is now being inclusive of cultivated meat. And similarly, so you have smaller startups that are doing that are sort of plugging into this sort of specialist role within the value chain to build this industry. But you also see very large corporations, multinationals, et cetera, that employ, thousands and thousands of people, carving out divisions within their entities to address, alternative proteins or cultivated meat as well. So I think when you start to see all of that sort of thing, those sorts of things come together, as well as increased public funding being more and more available, I think that is all good signs that this is going to last, and we're just really at the beginning phases.
Yeah, I agree. And also, we can see so many companies in Israel and so many opportunities, and each new development gives rise to a whole new industry. So, for example, when you identify a small challenge so many companies can rise and answer those new challenges. And I think this is very unexplored territory and can give rise to so many more advances, and also influences the industry of medical and pharma, give back the feedback to that.
Definitely. Certainly many adjacencies, both feeding into and spawning off of the cultivated meat field. All right, last question to our speakers before we wrap up today. Would love to just get a sense of whether you have any overarching career advice for cell biologists who are out there feeling maybe a little bit daunted about an entryway point into the cultivated meat space. Is there anything else that you have not said, a core piece of career advice that you'd like to impart on our audience?
I would advise them to talk with you guys and see the opportunities you're opening. And I know that GFI was very, very supportive in my transition to the cultured meat industry. So I want to say thanks and just be open to that.
I don't know. From my perspective, I think, you know, a lot of people think about job security or money or something like that when they're thinking about career selection and pathways, whether it's academia or industry. You know, you're in grad school and you're trying to figure that out. But I don't think enough people think about like what's going to provide them really the most joy in their work. And so if you can align your passion, like what you care about, with what you actually do for work, you're going to be much happier no matter where you land up. And so I think with cultivated meat, it intersects all these different things that a lot of people care about. So hopefully you can identify with one of those and align your career to address that. And I think you'll end up being quite happy with that decision.
Awesome. Thank you so much, Elliot and Roni. Thank you so much to Sharissa and the American Society for Cell Biology. And thank you to all of you for joining us today. Like Roni said, if you have curiosities, questions, and things that you'd like to discuss with us, please do check out our resources at gfi.org, and don't hesitate to reach out if we can be helpful in helping explore your pathway into the cultivated meat field. Thanks again for joining us today, and hope to be in touch soon. Thanks, everybody. Bye.